Hello, and welcome to today's Eco seminar. My name is Wout van der Bel. I'm a postdoc at Judith Menk at UBC. And before I introduce today's speaker, some small notes. So I'd like to remind all of you of our schedule for next week. So Monday next week, we'll have Judith Korb. On Wednesday, we'll have Rosemary Grant. And on Friday, we have Nicole King on our schedule. I'd also like to remind you that we'll have a Q&A session at the end of this seminar, and the link for the Slack where we have the Q&A can be found in the description of this video. And with that, I'd like to introduce today's speaker. So today's speaker is Professor Detlef Weigel. Detlef got his PhD from Tübingen and his postdoc at Caltech. He then got on faculty at the Salk Institute, where he's still associated today. He's also currently the director at the Max Planck Institute for Developmental Biology and adjunct professor at the University of Tübingen. Detlef has won many awards, including the Otto Bayer Award, the GSA Medal of the Genetic Society of America. He's also an elected member of the German National Academy of Sciences, as well as the US National Academy of Sciences. Also at the Royal Society of London and the American Academy of Arts and Sciences. So most of Detlef's work has focused on Arabidopsis thaliana. Detlef studies a wide range of topics in evolutionary biology, including plant-microbe interactions, hybridization, epigenetics, and even stem cells. I think today he's going to talk to us about epistasis and hybrid incompatibility. And with that, the stream is yours.
All right. Thank you very much. Walter, thank you very much also Elizabeth and Julia for inviting me, and thank you to the three of you for organizing this fantastic series. So before I begin, just disclaimers. I'm a deputy editor at eLife, something for which I'm getting paid. I also consult for KWS, and I'm a founder of Computomics, so to get that out of the way. I'll mention the people who did the work I'll describe as we go along, but I already want to specifically name people who are currently still in the lab: Christina Barragan, Talia Karasov. Talia is soon going to take up a faculty position at University of Utah, where she's starting her own lab. Lei Li, Derek Lundberg, and Manuela Neumann. And then people who recently left are Eun-Young Che, who is now an assistant professor at the National University of Singapore, and Dan Koenig, who is an assistant professor at UC Riverside. And you'll see photos of a few more folks as we go along. So I'm really, if I had to define what I am professionally, I would say I'm a geneticist. And genetics is what I love. I've even written an essay about this, why I love genetics, published in the Journal of Genetics. And I love genetics because they are these cool genetic phenomena. And perhaps the coolest phenomenon is epistasis. So without epistasis, genetics would be boring because it'd be entirely predictable. So here schematically shown some phenotype, here a genotype. So we have individuals of genotype A, we have individuals of genotype B, and then if we made a hybrid, A-B, we could predict the phenotype of the hybrid as the average of mom and dad. So hybrid strictly intermediate, no epistasis, boring. Now, there are interactions that play out differently. And here today is we have mostly evolutionary geneticists, and they'll know one definition of epistasis, but perhaps we'll also have a few developmental geneticists, and developmental geneticists actually use this other definition of epistasis. And so in developmental genetics, epistasis is typically defined as one parent being largely dominant. So the hybrid, in this case, would just look like parent B and completely mask the effects of parent A. Now, there's more interesting epistasis, actually, and that is when the hybrid exceeds the phenotypic value of either parent. And of course, one of the best known examples of that is hybrid corn. This is the basis of much of modern corn breeding. Now, there are these two different definitions, one where one parent is epistatic over the other, and this other definition where the hybrid has some new phenotype that is different from either parent. One might ask oneself, where do actually these different definitions come from? And I recently looked this up. And so it turns out the first mentioning of epistasis comes from Bateson, 1909, in his book Mendel's Principles of Heredity. And so he just described exactly this kind of situation, two different genotypes of different phenotypes, and hybrids look like either one or the other parent. And then he coined these terms epistatic and hypostatic. Hypostatic I hadn't heard before, but it's quite clear, obviously, what they mean. So where does this other definition come from, where epistasis is just any non-additive interaction between two genetic backgrounds? It turns out that also comes from one of the founders of modern genetics, no less than R.A. Fisher himself. And he was more thinking about how things like sex, male, female, for example, would globally change phenotypes. And when there was this global change in phenotype, he thought epistasy would be a good term for this. Now, this paper was reviewed, among others, by Punnett. And Punnett actually pointed out that this term epistasis had already been introduced by Bateson earlier. But Fisher just blew the reviewers off, and now we're stuck with these two definitions of epistasis. Okay, so I mentioned epistasis as evolutionary magic when the hybrids look much better, if you will, than either parent. But there's also the converse, epistasis as, if you will, evolutionary disaster. So where you have hybrid weakness or negative heterosis, where the hybrid is much less fit or much worse off than either parent. And this is really how we got into what we're studying today, these interactions within the immune system. That will become clear in the next few minutes. And so the person who got my lab into this is one of my first graduate students in Tübingen about 15 years ago, over 15 years ago, Janne Lempe, who is now at the Max Planck Institute for Plant Biology. And Janne was studying two strains of Arabidopsis thaliana. They both came from this village, Umkirch, not far from Freiburg, so not very far from Tübingen. And these two strains came from the same place, but they flowered at different times, as you can see here. One called UK1 for Umkirch, UK1, and the other one UK3. And so Janne thought, okay, since they come from the same place, genetically they're probably very similar. Maybe there's just a single mutation that made this strain here flower early or this one late. So let's just cross them together and see what happens. And so that's what she did. And a few weeks later, she came to me and said, well, this is very strange. I crossed these plants with each other, but these hybrids, they look really odd. So these are the two parents, and then similar age, the hybrid, and you see the hybrid is very, very small. So at the time, we were developmental geneticists, and we just stared at these plants. We put them on a scanning electron microscope. We thought it's just some, growth defect, some developmental defect. We couldn't quite figure out what's going on. So around the same time, Kirsten Bomblies, who's now a professor at ETH, joined us, and Kirsten started a series of further crosses between wild strains of Arabidopsis thaliana, and she found several similar cases. So we knew that this was sort of a phenomenon that one could study, that this was not just a one-off. So we stared at these plants, but we couldn't figure out what was going on. So we thought, okay, let's ask the plants what's wrong with them. So we extracted RNA and then looked at global RNA expression profiles. Back then it was microarrays. And these are three different crosses. And you see that these hybrids actually look quite different. So this one, is not too bad, whereas this one is teeny, 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 and this one is sort of in between. But even though the hybrids look fairly different in morphology, you see that these expression profiles are quite similar. So similar sets of genes induced and similar sets of genes reduced in expression in the F1 relative to either parent. And when we look at, what gene ontology, which GO categories were overrepresented, then the only ones that were related to immune response. And in plants, a typical outcome of activating the immune system, so plants being attacked by pathogens, they mount an immune response, a typical outcome is rampant cell death. And indeed, when we stained the leaves of these plants, we saw a lot of dead cells. So here, again, this dark blue stained with this stained trypan blue, you see all these dead cells, and we don't see much of dead cells in the parents. These are just the veins. So that fit. So that confirmed that some autoimmune defect apparently, and this autoimmunity reduced growth. So I don't have enough time to tell you everything about the plant immune system, but plants certainly prepare themselves for attack by pathogen, including microbes. And so we all know that dangerous microbes are everywhere. And that's also what the plants know. However, these plants, apparently, they just think they are microbes when there aren't any, because it's just from this cross that the immune system is being activated. So we think of these plants sort of as paranoid plants, plants which see pathogens, if you will, quote unquote, see pathogens, even if there aren't any. And so our hypothesis was that something that comes from one genome recognizes something that comes from the other genome as foreign or pathogen derived. And plants do have, similar to animals, receptors for recognizing intruders, for recognizing pathogens, for recognizing pathogen molecules. So we thought one genome might recognize some pathogen receptor, and then the other genome might contribute some random protein that somehow to this pathogen receptor looks like it is a pathogen protein. So, pretty simple hypothesis. And so this hybrid necrosis, as this phenomenon is called, because you have a lot of cell death, hybrid necrosis is sort of a thing. So when we went to the literature, we saw that there were hundreds of cases described by breeders and naturalists in pretty much any plant, except, curiously, not in maize. And this just shows in two examples from wheat. And actually, the genes had been mapped in the parents, and they were called necrosis 1 and 2. The interesting thing is that even though it was quite clear that this is probably always the same phenomenon in these different species, it hadn't actually been formally linked to immunity. So none of the previous authors had actually put 2 and 2 together, if you will, and none of them had said this looks like plants which are under pathogen attack, so autoimmunity. There was somehow this mental block that these immune response you only get when there's actually pathogen attack. So people apparently, couldn't imagine that you could get it just because of genetics. So we proceeded to map the causal gene, and it turns out that in this first case that we looked at, UK1, UK3, the genetics is simple, and that's typical for hybrid necrosis also in other species. So 2 clear genes, which we call dangerous mix, or DM. If anybody wants to look this up, this of course is a little bit tongue-in-cheek because it's also an abbreviation for Dobzhansky-Müller, and Dobzhansky-Müller incompatibility is something that is well known to students of evolution, of course. So 2 genes that interact epistatically, and the first gene that we were able to clone, identify, is this DM1, and encodes indeed an immune receptor of this NLR class, nucleotide binding site leucine-rich receptor class. And this is a structure published by Gigi Chai and Jianmin Zhu a little bit over a year ago of one such NLR immune receptor. And these genes are crazily diverse. So this is a small portion of the genome. And this is 3 different accessions or wild strains of Arabidopsis thaliana. This is the reference strain. You have 2 NLR genes that are very similar to each other. The UK1 strain, which has this good allele, so the non-causal allele, has also 2 genes, but they are both incomplete. So this one has a premature stop, and here just these fragments. And then the other strain, UK3, which has the bad allele, has just 1 complete copy, also a partial copy. But when you compare, for example, these 2 here, you find that there are 20% amino acid differences. So this is, of course, a crazy amount of difference, but very typical for NLR immune receptors, these very high difference. And that they are so different from each other has to do with them being often very old. And we'll get to that later to talk about the age of these alleles. So this was cool. So we went on and then cloned DM2, and we expected it to be just some random plant protein, as I explained earlier, that somehow looked to DM1 like it was perhaps a pathogen protein. And we were surprised when we learned that DM2 also encodes an NLR immune receptor. So it was two immune receptors interacting. And then sort of the mystery was resolved, and a student, Seb Tran, looked at the proteins, and they found these proteins actually in large complexes, both DM1 and DM2, and they can both homo and heterodimerize. And the way that we interpret this is on the background of what we know about how NLR immune receptors function. Again, they form these oligomers. The specific one for which we have this published structure, it forms this pentamer here. This is from a review by Tucken and Tamerling. So these NLR proteins have these nucleotide binding sites and enzymes. And enzymes go back and forth between different states, right? So here in the off state, ADP is bound, and you have this N-terminal domain, which is either a TER domain, a CC domain, and I thought that it's sort of folded over, and that's really confirmed by the structure here. And the NB domain touches the leucine-rich repeats here, and this whole protein is in a closed and off conformation. And then when pathogen effector comes in, you have the ADP is kicked out, and instead you get an ATP, and then you have this on conformation where now the business end of the molecule, either this TER or CC signaling domain, now is available for doing whatever job it has to do. And so our hypothesis from finding this direct interaction between DM1 and DM2 and then being in this complex was that normally you want to be in the off state and only go into the fully into the on state when there's a pathogen effector. But these things being enzymes, they go back and forth naturally between the off and the on state. But the off state should normally be preferred so that you don't signal in the absence of a pathogen effector. But when you bring together these two proteins, then very often they are in the on state even without an effector. So that was interesting. And we did know there were several cases, so we really wanted to know whether what we had found there was something more generally, that we often had interactions with immune proteins that caused these problems. And then Anyan Che came to the lab. It's about 10 years ago, and we had just started to resequence a good number of Arabidopsis thaliana accessions. And we had at that time 80 accessions, 80 wild strains resequenced from 8 different geographic regions, as indicated by these colors here. And Onyung said, okay, let's just go for it, and let's become Arabidopsis breeders and set up a dialogue. That is, take these 80 strains and cross everybody to everybody else. You see here she did an almost complete dialogue, so 6,400 crosses, over 3,000 unique combinations. And then wherever you see color is where you have hybrid necrosis, and they come in different flavors. So the worst cases, these are ones that die already as seedlings and which we cannot rescue by any trick. These magenta here ones or purple might appear on your screen. And then we have a lot of weaker ones, these blue cases here. You can see about 150 and these 6,000 crosses. So we think that this might be sort of a tip of the iceberg situation where these blue ones are ones that we can still, although often only barely see phenotypically, but probably there are more cases that are morphologically not apparent, but where the hybrids already have an elevated immune system status. And that's something that we've been trying to demonstrate directly for a while now. But long story short, this is really quite common. About 2% of F1s have hybrid necrosis, and you find it both within regions, so when you cross strains that come from the same region, as you find it between regions when you cross strains from this region to strains from other regions here. So at the time, as I said, we already had, we chose these strains because we had resequenced them. That made it easy to identify the causal genes. These are the 5 chromosomes of Arabidopsis thaliana, and then in color is this heat map of the distribution of NLR immune receptor genes in the genome. And there are something like 160 in the typical Arabidopsis thaliana strain. And you see right away that many of the causal genes map to regions where you have these NLR genes. And in several cases, we could show that indeed they were directly causal. And you have these different architectures. So this is one locus that we really like, DM2. This one was this where we first got this UK1 allele. So there you see there are 5 different hybrid necrosis risk alleles, and each of these interacts with some other region in the genome in some other strain. And then perhaps my favorite case is this one here, the interaction between DM7 and DM6, where we have 3 alleles at this locus and 3 alleles at this locus here, and they interact in this allele-specific fashion. RPP7 here, this NLR, and then RPW8HR, which is a known immune protein, but not an NLR. And so this is something that Christina Barragan in the lab has followed up on. So again, there are these 3 pairs of accessions. And when you cross Merck0 with KZ10, you get hybrid necrosis, but not when you cross Merck0 with Phase0 or Don0. And similarly, LARIC1, the 3 gives hybrid necrosis with Phase0, but not with KZ10 or Don0. In all 3 cases, when we mapped it, on this side, it mapped to RPW8 or HR. As I said, these are immune proteins. They share actually call domain with a certain NLR subclass, but they are not NLRs. And then RPP7, which is a family of NLR genes. And this locus here, the RPW8 HR locus, you see is also crazily diverse. So these are the major haplotypes that Christina could define. Here are 2 of the accessions where we have alleles that cause hybrid necrosis, and you see that there's a lot of sequence variation, copy number variation, and so on and so forth. And so this is the one that has a specific allele that we also find in Phase0 that interacts with a specific RPP7 allele. Here is another HR or RPW8 allele that interacts with another RPP7 allele, and so on and so forth. And so I was really fortunate because at that time, Lei Li joined the lab, and Lei's background was in the biochemical analysis of plant immunity and plant immune signaling. So he had really a very, very strong foundation in this area. And Lei said, this is really interesting. We know in the DM1, DM2 case, we have 2 NLR proteins that interact with each other, but here we have an NLR protein and non-NLR protein. What's going on here? And so he managed to show that in this case, also there's direct interaction between the causal RPP7 protein and the causal HR4 protein, and that this happens in an allele-specific manner. But what's important here is that the HR4 really acts as a ligand that induces complex formation of RPP7. So it's a bit different from DM1, DM2, where we have always these oligomers regardless of whether there is an apparent ligand. Here, it's really the HR4 protein that is required for RPP7 to oligomerize. So this is the combination of bad RPP7 and bad HR4, and you have this very large complex here of about a megadalton. Here you have the bad RPP7 and an innocuous HR4, and you see there is no evidence of such a large high molecular weight complex. Conversely, here is the bad HR4 and an RPP7 homolog that doesn't interact genetically, and again, there is very little, if any, evidence of such a high molecular weight complex. So how do we interpret what is going on here? It's not completely known what HR4 actually does, but the protein is similar to a protein in both fungi and also in vertebrates that are involved in non-self discrimination, and where these proteins can form pores in membranes. And so these pores in the membranes, they lead to cell death. And Ley could show that also HR4 is a very potent cell killer. So you can put it, for example, in yeast cells, you can put it into bacterial cells, and it will kill them. So there's evidence that it also functions like these other homologs, making a pore. So our idea then is that this is an immune protein that is being used by the plant when it recognizes a pathogen, and then is deployed to make these holes in the plant cell membrane. Now, what pathogens do is they typically inject their own proteins into plant cells to prevent immune response or attenuate immune response. So, hypothesis that these unknown pathogen effectors somehow will modify the HR4 protein so that they can't do their job anymore, that they don't go to the plasma membrane and make these pores. And now this is where the second layer of defense comes in, where plants have evolved these RPP7 receptors, and these RPP7 receptors, they will specifically recognize the modified HR4. They will not recognize the normal HR4, but only when it's recognized by a pathogen effector, and then they will trigger exaggerated defense. And this is all very much in line with what we know how these NLR proteins function. And then what we think is going on with, in this hybrid incompatibility case, that this HR4 phase 0 protein, that looks to the RPP7B protein like the pathogen effector normal HR4. So this one is obviously not modified by a pathogen protein, because this is just in this genetic cross, but it will still trigger activation of formation of this complex and activation of immune response. All right, so what we conclude from this, there's likely a wide range of direct interaction between NLRs. And it's what I find so fascinating about evolutionary biology, it's all about trade-offs. So from the plant perspective, one NLR protein is good because it provides resistance to some pathogen. 2 is even better because you're resistant to 2 pathogens, and so on and so forth. And we know that in a population, there's a very large number of different NLR immune receptors. I already told you they are very, very diverse. So why do typical Arabidopsis strains only have about 160 strains? Why 160 NLR genes? Why do they not have 500 or even 1,000? Well, we think the answer is the more NLRs you have, the more likely you are to form these heteromers, which signal even though when there's no pathogen. So there is this trade-off, more is better, but when you have too many, you run the risk of inadvertently activating immunity when you should not. And so this general idea is supported now by work from others, such as this paper from Frank Tucken's paper from my friend Sofian Kamoun, that many of these NLRs form these networks and a lot of intermolecular interactions going on. All right, so lessons: epistatic interactions in the plant immune system are common. Many translate into direct protein-protein interactions. We think that too much diversity at NLR and other immune loci can backfire. And finally, for aficionados of plant immunity, natural autoimmunity has actually revealed new molecular mechanisms in the plant immune system, just as an aside. All right, so I already alluded to this. NLR genes are crazily diverse, and this is from a paper we published last year. Annalena and Felix were in the lead here, where we had looked at the NLR gene complement in 64 wild strains of Arabidopsis thayana. We had initiated this before long-read sequencing of entire genomes was still expensive, so we had only looked at the NLR genes. But we had assembled about 13,000 NLR genes from 64 accessions. And this shows the presence of specific NLR genes in different accessions. And you see that most NLR genes are rare. So there are a few that you find in most or all accessions, but most of the NLR genes you find only in very few accessions. So that speaks to this diversity. You have a great pool of NLR genes, and then each accession selects, if you will, something like 150 or 160 out of this pool, and each accession has its own complement. So why are they so diverse? So this is something that Dan Koenig asked, although, in all fairness, we should say that Dan and the rest of the crew stumbled upon. So Dan was studying a genus of plants closely related to Arabidopsis thaliana called Capsella, and he was looking at 3 different species. Capsella grandiflora is this outcrosser here, has these large showy flowers, and there's a lot of diversity in the species. And then Capsella rubella is a sulfur, and we estimate that it became selfing something like 100,000 years ago, and it basically arose out of this outcrossing species. And you can see in this whole genome tree that there is very little diversity, and it nests completely within grandiflora. That makes sense. And then he also looked at this other sulfur here, which is further separated from the other 2, and we'll get back to that. So this species here comes from within the species, many transspecific alleles. That is sort of obvious, that what you would expect, because there's this common origin. What was not foreseeable is that these transspecific alleles are very non-randomly distributed. So this just shows chromosome 2. And what Dan found was that there were these very different regions of the genome. So there were large regions of the genome where you would have very little diversity within rubella, and it was highly differentiated between rubella and grandiflora. And the way we interpret this, that basically the species became selfing and a single chromosome was selected, and we're seeing essentially just a single chromosome with a few new mutations. Therefore, the species looks almost everybody in the species looks almost the same in this region. But then there are these other regions here where we have very high diversity within rubella, and there's very little differentiation. So the alleles that we see in rubella, we also see in the other species and in grandiflora. And then when Dan asked what's going on with these regions, they make up about 2% of the total genome, 21 regions he defined, that they are enriched again for immunity genes. So 9 overlap with NLR immune receptor genes, and 5 overlap with other immune genes. And several of these have a known function in thaliana in immunity. All right, so our interpretation was you could lose much of the diversity in the genome, but the immune genes were really special, that you cannot easily lose diversity at these key immune genes. Neat story, yeah, but it's n equals 1. It's really a so-so story. So needed some support from another system, and really very luckily, Barbara Neuffer and colleagues had discovered this other species here, Capsella orientalis, which occurs in a very different geographic regions. These are around the Mediterranean. This is from Central Asia. And this species here has also very little diversity, as you can see here from this whole genome tree. But even though there is very little diversity, so I think these were 16 individuals and only 70,000 SNPs total in these 16 individuals. When you look at 2 grandiflora individuals here, two of these individuals, they would be distinguished by 1 million SNPs, yeah? And these 16, only 70,000 SNPs. But out of these 70,000 SNPs, over 10% were actually shared with either grandiflora or with rubella or both. So again, extreme loss of diversity because of some very recent, very strong bottleneck, but some of these immune genes, really special, you can't lose diversity at these immune genes. And even more astonishingly, when Dan looked at, what were the genes where diversity wasn't lost, it was the same, very often the same genes as in Capsella rubella. So this is the frequency of transspecific alleles, and this shows the diversity in rubella. And you see that, the frequency of transspecific alleles in this other species in orientalis maps directly on where you have high diversity in rubella. And so this just is a nice visual example here what this looks like. So an ancient balanced polymorphism at one of these immune genes here called MLO2, where we walk along the genome. This is about 8,000 base pairs. can see we start over here. In blue is rubella, and red is orientalis. When we take a few individuals out here, you make a tree. The tree looks like you'd expect, that the two individuals go together and the blue individuals go together. But then when you get here into MLO2A, you see that all of a sudden this changes, where you have a red individual that goes with a blue individual, and again here you have a red individual that goes with a blue individual. You walk across this region, and here you come out again normally, where red goes with red and blue goes with blue. And so this is shown here also in this tree here. So at MLO2A, you can see that they are actually red, that is orientalis alleles, individuals that are very similar to rubella individual at this allele here, and similarly also at this allele here. And MLO2B is very different. So there the rubella and the orientalis individuals are nicely segregated. So there are no such sharing of ancient polymorphism. So if you walk along this region, this is what the diversity looks like. So in these two lines here, the dotted lines, this is the difference in base pairs between the same allele in the two species. So let's say here allele 1 from rubella compared to allele 1 from orientalis, okay? So this is difference between species, and you see this is something like 3%, and that's what you'd expect after 3 million years of evolution. And then here in purple and red, it's difference between different alleles in the same species, either within orientalis or within rubella. So difference between these and these here, or difference between these and these here. And you see this goes up to something like 15%. And so if 3% is like 3 million years, the separation of the species, or 2 million years, then 15% would mean that these are much, much older, that they are perhaps 10 million years old, these alleles. So these alleles are much older than either species. Okay, so this then is all very cool, but of course, in the end, it begs the question, why is it the same loci and often the same allele? So we have this interesting pattern. So here orientalis occurs here, grandiflora and rubella are over here. This is around the Mediterranean. This is 1,000 kilometers, thousands of kilometers away. Certainly very different pathogen pressures here than over here, yet it's the same immunity genes and same immune alleles that are being maintained in these populations to somehow persist. So how can we answer, where this diversity comes from? So there we really have to go to plants in the wild and ask what kind of microbes and other pathogens do we find on wild plants. And this is a work that was started by Talia Karasov and Manuela Neumann in the lab, and also Derek Lundberg, which I'm going to show on the next slide. And Talia and Manuela, in collaboration with the Kemen lab, specifically Juliana Almario, characterized diversity of pathogens in leaves of Arabidopsis thaliana around the area of Tübingen, and they did this at several sites over several years. And so what they found was that there is one specific taxon of Pseudomonas, and Pseudomonas are often plant pathogens, that was really common. And this taxon drives bacterial load. So this shows how many bacteria you have in a leaf. And if OTU5 is strongly abundant, then overall load is also high. So in line with what you'd expect, if this is pathogen, infects the plants, and then bacterial load goes up. Now, does that mean that this Pseudomonas taxon drives the evolution of Arabidopsis thaliana immune system? So perhaps it does. But what makes it difficult to understand is that we actually don't quite know who drives evolution of this specific Pseudomonas taxon, Pseudomonas viridiflava OTU5. So again, this is a work of Derek, whom I just mentioned. And Derek looked at several plants, including relatives of Arabidopsis thaliana and other plants. This is us here sampling Arabidopsis and other plants outside of Tübingen on this abandoned train track here. And what Derek finds is that this taxon is found on many Brassicaceae. So it is very common on Arabidopsis thaliana, but that's not the only place we find it. So this is what makes it difficult to understand immune system evolution in the wild, because you have you have a few specialist pathogens, but most pathogens are actually generalists. So it's a real challenge to map diversity in the pathogen onto diversity in the host. Yet this is what we are trying to do. So we initiated 2 years ago this project called Pathogen, short for pathogens on Arabidopsis, where we've been sampling diversity of Arabidopsis and the microbes and pathogens that live on Arabidopsis thaliana throughout much of Europe and even into Asia. And with that, I'd like to conclude, and I want to conclude with this quote from Rosemary and Peter Grant, and really very befitting that Rosemary will speak next week from their wonderful book, How and Why Species Multiply. It's, I think, the last sentence in their book: nothing in evolutionary biology makes sense except in the light of ecology. So we're trying to understand these evolutionary patterns, but we can only understand them if we actually go out into the wild and ask, what does it look like there? And so I want to conclude and leave you with some of these impressions, what it looks like when you sample Arabidopsis thaliana in the wild. This, for example, is Arabidopsis growing on the coast of the Baltic Sea. Here's Arabidopsis thaliana in an abandoned cloister in Ireland, here in southern Italy under cactus and so on. So you get a nice impression that Arabidopsis thaliana, it's a real plant, but it's also adapted to very different environments, and thus it does make a very nice evolutionary and ecological model. And with that, I'm happy to acknowledge especially support from Max Planck. I say thank you to all of you who are online listening to me and hearing about the work of my group. And I'm happy to take questions. Over to Walter.
Thanks so much. That was an amazing talk. I guess I can start with one of my own questions. So,
Walter, should I do the stop screen share, or?
It's fine either way.
Okay.
Yeah. So it's quite surprising to me that in this diallelic cross, you find like 2% of crosses are affected, right? Seems like a lot. So do you have any idea of like whether you have similar widespread problems outside of plants, or how common is it even like within plants more broadly?
Yes. So that's very much spot on, Walter. So first of all, within plants, one of the questions that I'm often asked is Arabidopsis thaliana is a selfer, but it actually does outcross quite a bit. So when we look at the wild plants, we see that outcrossing is about 10%. And so when you look at, for example, linkage disequilibrium, Arabidopsis sort of looks like humans. So the effective outcrossing rate is similar to humans. So Christina, one of my students whose work I already mentioned, she has been looking at an outcrossing relative, and she has found that actually deleterious interactions are quite common. We haven't been able to pinpoint the genetic causes of any of these deleterious interactions she has found, but she found that they are much more common, at least when you grow plants in the lab, than what you would perhaps naively expect. It's within in outcrossing species, it's not, it's more difficult to observe, because you have things that are segregating. Everybody is heterozygous, so not all the hybrid offspring will suffer from this. But there's, for example, very nice work in Drosophila. There's now also work in C. elegans. So these deleterious epistatic interactions within species, they have turned out to be much more common than I think many people would have thought even 15, 20 years ago. And so in humans, that's actually something I find really quite interesting. So how many cases of infertility, for example, are because of epistatic interaction? There's some indication that actually also the immune system might contribute to some cases of infertility in humans.
Fascinating, thank you. So we'll move on to some questions from the audience. So can you say anything about what general factors might predict if hybrid necrosis will occur? Like, could it be related to the degree of sequence differences between parents, for example?
Yeah, so that would be a dream. So it would be a dream if we could look at the NLR complement of two strains and predict which NLRs are likely to make these complexes, which don't work well. So unfortunately, because the diversity is so excessive, I think it has been pretty difficult to really classify and systematize things, but that certainly would be our ambitions. But so I mentioned in these 65 accessions, we looked at NLR diversity, and I mentioned it was 30,000 genes, but that's of course, alleles, but they collapsed into thousands of genes. So then when you think about the interaction matrix, that's millions of interactions. So hopefully with a better understanding how they work at the molecular biochemical level, at some point that will become possible. We have, of course, looked where in the protein the differences are, and it's normally in these leucine-rich repeats, which are very variable. So that sort of makes sense, but we are very far from predicting anything.
It's going to be a lot of molecular biology then.
Yes. Mm-hmm.
So are there any differences in the hybrid behavior if it grows in different environments? So for example, parent 1 native environment versus parent 2 native environment, etc.
Yeah, so we have seen hybrids occur in the wild. What we know in the lab is that this phenomenon is very much temperature dependent, and that's typical for many immune reactions, not just autoimmune reactions. At a higher temperature, they often go away, and this is also what we have seen. So when we grow them in the lab at temperatures that are typically above what plants experience outside, they will actually survive. So in principle, what that would mean is that if hybrids would grow in an environment that's more typical, say, for a Mediterranean climate, they ought to be more likely to survive. We haven't formally looked at this, but we have actually seen some of these hybrids which look really bad in the lab. We have seen them growing outside in Spain.
All right. Could there be interactions between the NLRs from each parent in the hybrid?
Yeah, so that would be sort of that's also a very astute question. So that sort of could be the converse. So I have emphasized that you bring together NLRs from the different parents, and then bad things happen. But it's very much conceivable that you actually have at a very much sublethal threshold that you actually have NLRs in one parent. They interact with each other and actually do activate the immune system to a certain extent, reduce growth. And then when you make a cross, you dilute that interaction and you reduce immunity. And it's something that I would love to find, and we have tried really hard to find this, and technically that has really been challenging, but I would absolutely bet that this probably exists. So that that hybrid necrosis could be sort of the opposite of hybrid vigor, and that you could get hybrid vigor by diluting hybrid necrosis.
Yeah, do you know of any evidence that hybrid vigor, there's like also a strong role for the immune system there? More generally.
Yeah, so as I said, so we'd love to find this. Unfortunately, it's much easier to find the ones that have the activated immune system than the one that have the reduced immune system, because it will be more difficult to test, basically, because normally the immune system is off. So you would have to really do a lot of pathogen testing, which is more challenging. But conceptually, and we actually worked with Michael Derbley, whom I'm sure in Vancouver, and so he has shown theoretically that one would very much expect anti-hybrid necrosis. So he has published a lovely theoretical paper that made that point.
Thank you. Are the genes which cause heterosis in the hybrids also limited to certain categories in Arabidopsis?
so I think this goes more in hybrid vigor, when if I understood correctly. So hybrid vigor heterosis, that's one that remains one of the super big challenges in plant biology. And in Arabidopsis, we have looked at hybrid vigor, and there it seems that when we can pinpoint genes, that often few genes are involved. The same is true in tomato. There's lovely work from Zach Lippman, who has shown very few genes can underlie hybrid vigor. But in corn, it's clear that it's many, many different genes. So probably for hybrid vigor, there's not one size fits all to begin with in terms of genetic architecture. And then the things that we have found in Arabidopsis, they were all over the place. And when you look at tomato, I think it's also similar, that they tend to be all over the place.
Mm-hmm. Is hybrid necrosis similar to what happens when two different species show reinforcements against hybrids?
Yeah, so I mean, we have, it's certainly something that we would expect. So the way we think about this, that you have these negative interactions between these NLR genes because you have combinations that have not been tested in the wild, or combinations that, genes that normally don't co-occur. And obviously when a species separate, then, NLR genes will evolve on separate tracks. And therefore, it becomes much more often, much more likely that you could get such deleterious interactions. It hasn't been formally tested whether it's more common between species, but there are certainly cases of hybrid necrosis between species. One should also point out, again, that's one thing that I find so fascinating about evolutionary biology, that there are many different ways to get negative interaction. And so tomato, for example, it's very clear that, so there's also some hybrid necrosis in tomato, but in tomato, you get hybrid sterility as pretty much the first thing right away. Whereas in Arabidopsis thayana, we have never observed hybrid sterility. So again, it's dangerous to conclude from one, or to extrapolate from one species to others. And again, that's I think why we all love evolutionary biology.
It keeps you busy, huh?
Yep.
Were there significant differences in time of observation of autoimmune phenotypes in hybrids, or they all showed reduced growth or necrosis right from germination?
Yeah, so that does differ. So the strengths of the effect certainly differ. So we have some that germinate relatively well, and some of them, there's this overgrowth phenomenon where you have apparently immunity going on, but then the plant partially can outgrow it. But then we have some that die as seedlings, where the true leaves never develop. And so this is another case that Christina has been working on, one of these really super strong cases. It's really quite fascinating, because when you look at the gene expression profile, you see that pretty much all the NLR genes just go through the roof. So they are transcriptionally activated, to the nth degree, and very clearly that's what kills the plants. But there seems to be quite some diversity, and also when you morphologically look at the plants, they look quite different.
So you partially answered this already, but how stable are epistatic interactions under differing environmental factors, especially in plant breeding programs where gradually increasing environmental temperatures are reported?
Mm-hmm. Yeah, so the wheat case is actually really super interesting. So there are these necrosis genes, and so they provide benefits in terms of fungal resistance, and they have increased in frequency within the population. So they are certainly frequency shift. Now, typically, you would not grow hybrids in the field that show any of this, so they get eliminated. It's practically more of a problem for breeding. So as I said, in wheat, you have these two different genes, and you basically have one pool of parents that have one gene, you have another pool of parents that has the other gene. There are very few lines left that have neither of these genes. and then it becomes more and more difficult to cross them to each other. But in the field, when you grow plants for, growing food and whatnot, it wouldn't be an issue, because these plants you would never sow out in the first place as a crop.
So in a more natural environment, then, if we think about climate change and increasing temperatures, is that going to really affect how likely crosses are to persist?
Yeah, so I think it's more that I think the more important thing is going to be that temperature has a huge effect on the immune system in general, so that high temperature it stresses weaken the plant's immune system in general. I think it's a plausible scenario that, as temperatures become warmer, that perhaps there are some hybrids that are all of a sudden fitter than before, and that could affect gene flow. That's a very plausible scenario. But I think the bigger challenge for the plant is just in general that the immune system might take a hit.
Okay, great. I think that's the questions we have for now.
Awesome.
So with that, I would like to remind people to for Monday's seminar with Youth Corp, which I think will be about aging in termites. And with that, I'd like to thank again Professor Weigel for your time and your effort. And I hope to see you all back on Monday. Goodbye.